Hi students. So welcome back to another new session. So today we'll be dealing with the topic structure of DNA and packaging of DNA. Okay. So this is from the chapter from our class 12 NCRT text. Okay. And this topic belongs to chapter number 5, and you all know it is molecular basis of inheritance. Okay, so we have this topic coming under this chapter. So very important chapter and very important topic in examination point of view for both NEET, okay, then CUET, and for your class 12 boards. Okay, so for many of them, this will be a very new topic because you will not have any continuation from your class 11 or 10. You might have studied about the genetic material DNA, its full form. But here in this topic, we'll be studying in detail about what the structure of DNA is, how is DNA kept safely inside the nucleus of our cell, etc. So let's start the session. Yes. So the first what we need to study is about DNA. What exactly is DNA, right? So it's a long polymer. Okay, it is a polymer of deoxyribonucleotides. Okay, so DNA is what? It's not a monomer unit, but it is a polymer. Polymer means what? It will be a continuous chain made up of small monomeric units. So here the monomeric units are deoxyribonucleotides. Okay, deoxyribonucleotides. So these will be the monomeric units in case of DNA, which is a polymer. So we'll be studying in detail about this deoxyribonucleotides. Okay, its structure, everything. Okay, and as we all know, all these, that is nucleotides as such. Okay, we have two types, ribonucleotides and deoxyribonucleotides. Both comes under what? Nucleic acids, right? So you all know the full form of DNA. So what is the full form of DNA? Deoxyribonucleic acid, right? So I'll just write it down here. It's deoxyribonucleic, then acids. Okay, so this is the full form of DNA. Okay, so it's a polymer, as I said, made up of monomeric. We'll be dealing very shortly. Okay, the length of the DNA is usually defined as number of nucleotides which consist of a monomeric unit. Okay, so then the length, okay, this polymer's length or the DNA's length is usually defined by how many nucleotides we have in them. Okay, it is characteristic of an organism. Okay, what is characteristic? The length of the DNA or the number of the nucleotides present in the DNA is characteristic of an organism. That means it will be unique for an organism. For example, the bacteriophage lambda. So what is this bacteriophage? Bacteriophage is actually the viruses that affect bacteria. Okay, we call them generally as bacteriophage. So one kind of virus which affects bacteria is bacteriophage lambda. And bacteriophage lambda has how many base pairs? 48,502 base pairs. Okay, that is actually unique for bacteriophage lambda. That's what I said. The number of nucleotides present and therefore the length of the DNA is characteristic of an organism. That is, that particular factor is unique for an organism. Okay. E. coli has 4.6 into 10 raised to 6 base pairs, right? So E. coli is a Bacteroideaceae coli. If you look into the E. coli's genetic material, it has how many base pairs? 4.6 into 10 raised to 6 base pairs, which is again unique to E. coli. Okay, and so that we covered. The next one is the haploid, right? The haploid content of human DNA. Okay, haploid content of human DNA means our own DNA, we humans DNA, right? So, how many, what is the number of base pairs found? 3.3 into 10 raised to 9 base pairs. Okay, we can find 3.3 into 10 raised to 9 base pairs. So this means this is just the haploid content. So normally our cell is diploid in nature. So students, I hope you know what haploid and diploid is. So if you say that a cell is in haploid condition, okay, haploid condition means how do we generally denote it? We denote it as N, right? N condition. Okay. And when we say that it is diploid, right? When the cell is in diploid condition, we represent them as 2N, right? So here, if we say that the haploid content of human DNA is 3.3 into 10 raised to 9, then the diploid content is 2 into 3. That is, just multiply it with 2, right? Then how much do we get? 6.6 into 10 raised to 9. Okay, so that many base pairs are found in what? In human DNA. So what I'm trying to say here is that, as I told in my third point, the number of the number of nucleotides present in the DNA, therefore the length of the DNA is characteristic for an organism. If you take the case of bacteriophage lambda, it has 48,502 base pairs. If you take the case of E. coli, it has 4.6 into 10 raised to 6 base pairs. Okay. Now let's go into the structure of the DNA. So I told DNA is what? It's a polymer, right? So polymer of what? Deoxyribonucleotides, or you can otherwise generally we can call them as nucleotide, polynucleotide chain. Okay, we can generally call them as polynucleotide chain. So when you consider the structure of a nucleotide has three components. Okay, a nucleotide has three components. So here, it's written here, there is a nitrogenous base, a pentose sugar, and in case of RNA, it will be a ribose sugar. And in case of DNA, it will be deoxyribose sugar and a phosphate groups. So this is the components or the three components of a nucleotide. So we have a nitrogenous base, a pentose sugar. Again, pentose sugar, we can find two types. One is ribose sugar and the other one is deoxyribose sugar found in DNA. Then we have what? Then we have phosphate group. So these are the three components of a nucleotide. Okay. Then when you take the case of nitrogenous base, that is, now if you pick out each components of these nucleotide, first if you consider the nitrogenous bases, there are two types of nitrogenous bases, purines and pyrimidines. Okay, we'll be studying shortly the structure of purines and pyrimidines and all. So we have two types of nitrogenous bases. One is purine and the another one is pyrimidine. Okay. Again, when you consider purine in our genetic material or nucleic acids, the purine nitrogenous bases are of two. One is adenine and the other one is guanine. Okay, so adenine and guanine. So students, please be careful. Nitrogenous bases, we have two types, purines and pyrimidines. And under purines, we have two coming in, that is adenine and guanine. And under pyrimidines, we have what? Again, three coming in, that is uh, cytosine, thymine, and uracil. Okay, we have three coming in, cytosine, thymine, and uracil, right? So you have to learn this, you have to keep it by heart. Purines, you have adenine and guanine coming in, and in pyrimidines, you have three nitrogenous bases, cytosine, thymine, and uracil. Okay. Now, cytosine, when you take the case of pyrimidines alone, okay, so this is a pyrimidine, cytosine. Cytosine is common for both DNA and RNA. So, when you study about nucleic acid, so nucleic acid is a biomolecule that you have, I think you have studied in class 11. So, when you take nucleic acid, I'm representing it as Na, okay, nucleic acid, you have DNA as well as RNA coming in. Okay, DNA and RNA. So we'll be studying the key differences of both of these. So one of the differences present in its pentose sugar. Okay, in its pentose sugar. For both, in case of sugar will be different. So in here, in case of deoxyribose. Okay, it's already written in the slide, deoxyribose for DNA. And for RNA, the sugar's name is ribose. Okay, ribose sugar. Okay, this is one of the difference. Okay, it is based on the presence of an OH group in the second carbon of the pentose ring. Okay, then another difference is in the nitrogenous base. Okay, DNA has purines, adenine and guanine. When it comes to pyrimidines of DNA, we have cytosine coming in, and instead of thymine, and cytosine and thymine. Okay, in DNA we have purines, that is adenine and guanine, and when you consider pyrimidines for DNA, we have cytosine and thymine coming in. But when you take the case of RNA, and when you consider nitrogenous bases, purines, both adenine and guanine is present in RNA. Okay, both adenine and guanine is present in RNA. But when you take the case of pyrimidines, thymine is absent, or we can say that instead of thymine, uracil is present. So for when you take the case of both DNA and RNA, you can find purines, that is adenine and guanine. And in case of pyrimidines, instead of thymine, you have uracil. So in RNA, the nitrogenous bases, okay, nitrogenous bases found are what, especially particularly the pyrimidines, it is cytosine, cytosine and uracil. Clear? Okay. So that's what I have written here. Cytosine is common for both DNA and RNA. Thymine is present in DNA, but it is absent in RNA. Okay. Now, this is the structure of purine. Okay, so we have the structure of purine here. It's a 7-membered heterocyclic compound. Okay, so you have 2 rings attached here, a 5-membered ring, and 1, 2, 3, a 4-membered ring. Okay, a 5-membered ring, sorry, 6-membered ring and a 5-membered ring. Okay, and these are the structure of adenine and guanine. Okay, and when you take the case of pyrimidines, okay, again we can see thymine and cytosine. Thymine only found in DNA, and this is the uracil. This is the general structure of pyrimidine. Pyrimidine is a single ring compound. Okay, so next, yes. So let's see how these components are linked and how a nucleotide is formed. Okay. So a nitrogenous base is linked to the OH of the first carbon of pentose sugar through N-glycosidic linkage to form a nucleoside such as adenosine or deoxyadenosine. So when you study the structure of a nucleotide, so we have the pentose sugar. Okay, so this is the pentose sugar. Okay, our 5-membered ring. This is the pentose sugar. Okay, so to this pentose sugar, the OH in the first carbon. So this is the first carbon. This is the second carbon, third carbon, fourth carbon, and we have a fifth carbon coming here. Okay, so in the first carbon's OH group, there we can find the nitrogenous base, and it is linked through, or the nitrogenous base is linked to the OH group of the first carbon of pentose sugar via N-glycosidic bond. So we have what, we have N-glycosidic, bond. Okay, N-glycosidic bond. So this bond's name is N-glycosidic bond. It is very important. They usually ask this for exams. Okay, so this is basically this whole structure, that is sugar with nitrogenous base. We call them as nucleoside. What it is? Sugar with nitrogenous base, we call them as nucleoside. So if we have the nitrogenous base adenine coming in, okay, if the nitrogenous base is adenine which is attached to the pentose sugar, okay, we call it as adenosine. Okay, in case if the sugar is ribose, so I'll just mention here. So we have this ribose sugar. Okay, ribose sugar. To the ribose sugar, what is attached? adenine is attached, okay, a nitrogenous base is attached, and via N-glycosidic bond. So we call them as adenosine. Okay, we call it as adenosine. Okay, generally we call them as nucleoside. Okay, now another, that is the next type of nucleoside formed is with deoxy deoxyribose sugar. We have got an attached adenine nitrogenous base, and we call this as deoxy adenosine. Okay, so I hope you are clear with this. So nucleotides, we can name it based on the sugar, based on the type of sugar to which the nitrogenous base is added. That can be either a ribose sugar or it can be a deoxyribose sugar. So similarly, based on the nitrogenous bases added and the kind of pentose sugar to which this nitrogenous base is added, we can call them as guanosine or deoxyguanosine. So in this case, the nitrogenous base added is guanine. Okay, guanine. Then it can be cytidine or deoxycytidine. So here the nitrogenous base added is cytosine. Okay, and in this case, you can call them as either uridine or deoxythymidine. So uridine you can actually find in RNA. Okay, you can find in RNA, and deoxythymidine you can see in DNA. Okay, is it clear? Right. So let's see. So in this slide, we studied how a nucleoside is formed. So next we learn how a nucleotide is formed. So when a phosphate group is attached to the OH of the 5' carbon of deoxythymidine through a phosphoester linkage, a corresponding nucleotide is formed. So till this much we saw in the previous slide. So to this compound, to the 5' carbon, okay, to the 5' carbon, when a phosphate group is attached, okay, via what bond? Phosphoester linkage. Via phosphoester linkage, we call it as what? We call it as nucleotide. Okay, we call it as a nucleotide. So here they have simply just mentioned to the deoxythymidine. That means thymine group will be the nitrogenous base found, and through the phosphoester linkage, that is a phosphate group will be added to the 5th carbon of the pentose sugar via phosphoester linkage. Okay, phosphoester linkage, and then a nucleotide will be formed. Okay. Yes. So let's see how the linkage is happening and everything of that sort. So two nucleotides are linked through 3' and 5' phosphodiester linkage to form a dinucleotide. So we said DNAs are actually what they are, polymers of nucleotides. So more than one or two nucleotides, they have to join together, right? So how do they join? Two nucleotides are linked through phosphodiester linkage. So it's 3' 5' phosphodiester linkage. Okay, and they form a dinucleotide. More nucleotides can be joined in such manner to form what? A polynucleotide chain. Okay, so more nucleotides are added. So if two are added, it becomes dinucleotide. If three are added, it becomes trinucleotide. Later, it forms a polynucleotide chain. Okay, a polymer thus has at one end a free phosphate moiety, that is a 5' end. Okay, it has a free phosphate end, and it is referred to as the 5' end of the polynucleotide chain. So if you take a polymer, on one side it will have what? A 5' end, just denoting on which carbon the phosphate group is added. And on the other end of the polymer, so if you take, if I'm just showing you an example, so this is a deoxyribonucleotide unit. So we have units like this coming in. Okay, we have units like this coming in. So this end will be 5' end, and this will be what? 3' end. Okay, so one end we have the 5' group, and the other end we have the 3' group. So at the other end of the polymer, the sugar has a free OH of 3' carbon group, which is referred to as 3' end of the polynucleotide chain. So we call them as what? We call them as 3' end of the polynucleotide chain. That is this end. Okay, and this is the 5' group. Okay. The backbone of the polynucleotide chain is formed due to sugar and phosphate. So when you take the case of the polynucleotide chain, it's stabilized, or the backbone is formed through what? The sugar and the phosphate groups. Okay. Then nitrogenous bases linked to the sugar moiety, they project from the backbone. So the backbone is formed by sugar phosphate. Okay. If you take the case of a polynucleotide chain, the backbone is formed by sugar phosphate, whereas we have nitrogenous bases which is linked to the sugar moiety projecting from the backbone. Okay, projecting from the backbone. So that will be seen as a projected moiety. Okay, I hope it's clear how the structure goes. So this is the polynucleotide chain structure. So this is the 5' phosphate. So this is attached to the 5' carbon. Here we have the fourth one, here is the third carbon, second carbon, first carbon. So to the first carbon, as I said, to the first carbon, we have the nitrogenous base attached through glycosidic bond. Okay, glycosidic bond. Then onto the fifth carbon, we have the phosphate group. So we call them as 5' phosphate. The 3' OH group is attached to the, again, the phosphate group of the second deoxyribonucleotide. That's third carbon, right? The third OH group is again linked through phosphodiester linkage to the fifth carbon of the next ribonucleotide. So this is the phosphodiester linkage. This is the, sorry, spelling. Okay. So this is the phosphodiester linkage. Okay, this is the phosphodiester linkage, right? And this linkage, right? So I am talking about this one. This is the phosphodiester linkage. So what is this linkage called? This is just phosphoester linkage. Okay, this is just phosphoester linkage. Clear? So we may have phosphodiester as well as phosphoester linkage. So make sure you don't confuse it with both. The phosphate group is attached to the nucleoside via phosphoester linkage, whereas two nucleotides are attached together through phosphodiester linkage. Clear? Yes. So with this, we have done with DNA. I hope everything is clear. So we started off with discussing DNA as a polymer of deoxyribonucleotide. Then we told about the basic, the base pairs, how it is characteristic. Then we told about different components of a nucleotide, the nitrogenous base, the pentose sugar, the phosphate group, everything. The two types of nitrogenous bases, what is present in RNA, what is present in DNA, the structure of purines and pyrimidines. Students, please make sure you learn this because it's very important. Okay. Then we told about how a nucleoside is formed, how a nucleoside becomes nucleotide. Then we told about the sugar phosphate bond, the 3' 5' phosphodiester linkage, all of that. Okay. So next we are going into RNA. So this much, that we were studying about DNA, right? Till now we were studying about DNA. So now we are going into RNA. So let's learn this. Every nucleotide residue has an additional OH group present at the 2' position in the ribose. Okay, that is, when you take the case of RNA, we told that the pentose sugar found is ribose sugar, right? Ribose sugar. And in case of DNA, we told it is deoxyribose sugar. So how is that sugar different? Sugar is different in just one position, that is in the second position. Okay, in case of RNA, when you look onto the pentose ring, at the second position they have a OH group in case of RNA. But in DNA, in deoxyribose sugar, there is no OH group in the second position. Okay, in the second position we cannot find any OH group. So that is the only difference, not OH group, just the oxygen part. That's why we call them as deoxyribonucleotides. The O group will be lost. You can only find hydrogen in that specific area, right? Then the second difference is uracil is found in place of thymine. So in RNA we can find uracil, not thymine. And 5-methyl uracil is another chemical name for thymine. So when you another name for thymine is 5-methyl uracil. So to the uracil, if you add methyl group at the 5th position, then we get a thymine. So that is the idea. Okay. So here we have in one side we have what? We have the DNA's nitrogenous bases, and in the second side we have RNA. So DNA is always double stranded. Okay, DNA is always double stranded, and we can find the pyrimidines and purine nitrogenous bases found here. Okay, and in case of RNA they are single stranded. Very important point. Okay, they are normally seen as single stranded. And when you look on to the nitrogenous bases found, instead of thymine you have the uracil. Okay, and cytosine, adenine, guanine. Then this the second difference that you have to study is when you take the case of pentose sugar. Okay, pentose sugar of DNA and pentose sugar of RNA. So in case of RNA, you have what? In the 2' position you have OH. Okay, and in case of DNA, okay, in case of DNA, you have no, you don't have that oxygen group, or there is no O present. You just have what? 2' H in the second position of the pentose string. Okay, so that you have to be careful. You need to understand the basic difference in the pentose sugar as well as in the nitrogenous base. Yes, so next we have some facts about the structure of the DNA. First is DNA is an acidic substance and it is present in the nucleus. So chemically DNA is acidic in nature and they are negatively charged. Okay, so this also you need to keep in mind that they are negatively charged molecules. Okay, and they were first identified by whom? Frederick Miescher. Frederick Miescher identified it and he called it as nuclein. So this is how the discovery of DNA goes. So it was first identified by Frederick Miescher and he named it as nuclein, and DNA is an acidic substance and it is negatively charged. Okay, James Watson and Francis Crick, based on the X-ray diffraction data, proposed the double helix model of DNA. So who proposed the double helix model of DNA? It was James Watson and Francis Crick. So they are the two scientists, okay, who based on the X-ray diffraction data, they proposed what? The structure of the DNA is double helix. So that's very important and very important discovery, that DNA has a double helix model. Okay. Based on the observation of Chargaff for double-stranded DNA, the ratios between adenine and thymine and guanine and cytosine was also found. That is Chargaff, a person, okay, he observed this double-stranded DNA and he said that the percentage of adenine is equal to the percentage of thymine in case of a double-stranded DNA. So you have to make sure that this point, double-stranded DNA, is very important. So this ratio of percentage of adenine becoming equal to percentage of thymine can only be seen in a double-stranded DNA, not in a single-stranded DNA. Then he also found that the concentration of guanine, okay, concentration of guanine is equal to concentration of cytosine. Okay, so if you take the ratios, that is A by T, it will be equal to 1. Similarly, G by C will be also equal to 1. Otherwise, you can also write this as A plus G by okay, A plus G by T plus C is also equal to 1. Okay, right. Next, the base pairs are complementary to each other and therefore the sequence of bases in one strand is known, then the sequence in the other can be predicted. So this is again another important fact, that is all the base pairs that we were talking about, the nitrogenous bases, they are complementary in nature. Okay, we'll study how they are complementary. That is, I'll just say, for example, if you take the case of you have adenine base pair here, then we have thymine, guanine, cytosine. Then this is the order of nitrogenous bases in one of these strands. Okay, if you know this, you can actually find the sequence on the opposite strand because they are complementary in nature. So adenine is complementary to thymine, thymine is complementary to adenine, guanine is complementary to cytosine, cytosine is complementary to guanine. So in this way, you can identify the sequence in the opposite strand. If each strand from a DNA act as a template for the synthesis of a new strand, the two double-stranded DNA produced would be identical to the parent DNA molecule. Okay, so you might have heard about what replication of DNA, right? Replication. So I'll just 1 minute. I'll just write it over here. So we have a double-stranded DNA. Okay, we have a double-stranded DNA. So this DNA undergoes denaturation and become two single-stranded DNA. Okay, and what happens? Replication is like a complementary strand. Okay, a complementary strand complementary to the parental strand will be formed. Okay, parental strand will be formed, and this will be exactly same as what the parental strand. Okay, I hope you got it. Okay, so for the replication, prior to replication, there will be a denaturation of this double-stranded DNA, and a single strand will be formed, and a complementary strand will be formed for this, like this. And these two daughter strands or these two daughter double helix DNA will be similar to the parental DNA. Okay, I hope it's clear for you. Right. So next we go on to the salient features of double helix structure of DNA. This is very important. We have 5 points coming under the salient features of double helix, and you need to learn all of that. More than by hearting, you need to understand and learn what exactly they are telling us. Okay. So the first feature is it is made up of two polynucleotide chains where the backbone is constituted by sugar phosphate and bases project inside. Okay, so we already told about that the backbone is constituted by sugar phosphate, right? It has a sugar phosphate backbone. And what about the bases? The bases project inside, right? We have the bases inside along, that is they project from the sugar moiety. Two chains have anti-parallel polarity. It means that if one chain has polarity 5 prime to 3 prime, the other has 3 prime to 5 prime polarity. This is very important. Okay, so here if you take the case of this particular strand, so this is when you take the double stranded, this is the first strand and this is the second strand. So if one strand is 5 prime, so it starts here, right? If it's 5 prime, 3 prime, the second strand will be 3 prime, 5 prime, right? That is what you mean by anti-parallel polarity. Or simply if I'm drawing two strands like this, if one is 5 prime to 3 prime, then the another one is 3 prime to 5 prime. Okay. The third salient feature is the bases in two strands are paired through hydrogen bond forming base pairs. Okay, adenine, it forms two hydrogen bonds with thymine from opposite strand and vice versa. So we already told about the complementation, right? So adenine always complement with thymine. So they complement via what? They complement via hydrogen bonds. So adenine is bonded to thymine via two hydrogen bonds. Okay, so adenine is bonded to thymine via two hydrogen bonds. And guanine is bonded to cytosine via three hydrogen bonds. Okay, so you need to learn that adenine is complementary to thymine and vice versa. Okay, thymine is complementary to adenine. Guanine is complementary to cytosine and cytosine is complementary to adenine. And they are linked through hydrogen bonds in case of adenine and thymine via two hydrogen bonds, guanine and cytosine via three hydrogen bonds. Okay, so as a result, when you look onto this, purine comes opposite to pyrimidine, or purines are always complementary to pyrimidine. and this generates approximately uniform distance between the two strands of the helix. So when you take the case of this, what it is, we can find, because complementation can be seen like this, you can see, you can have approximately equal distance between the two strands. Clear? So, so this is what I was saying. So in this, you have a ladder-like thing. You have two strands coming in. Here the adenine. Okay, adenine complements via two double bonds with thymine and thymine with adenine. Okay, and you can see an equal spacing also because of these base pairing. Okay. The next important point is the two chains are coiled in right-handed fashion, right? They are coiled in right-handed fashion, and the pitch of the helix is 3.4 nanometer. Okay, so what is pitch? So let me first draw the structure of the DNA, the double helix model. It looks like this. Okay, so pitch means the distance of one turn. So if you take the case of this DNA that I have drawn, it has, this is one turn, this is the second turn, this is the third turn, and this is the fourth turn. Okay, so the distance of one turn is what we call as pitch. Okay, what we call as pitch, and it is how, what is the distance, or what is the length of that one turn? It is 3.4 nanometer or 0.34 angstrom. Okay, you need to learn this, you need to keep it in your mind. Okay, we'll be dealing with questions regarding this. So pitch of a DNA, or the length of one turn, is what? It is 3.4 nanometer or 0.34 angstrom. Okay, and in this turn, okay, if you count the number of base pairs found in each turn, you have approximately 10 base pairs coming in. Okay, you have 10 base pairs coming in one turn or in pitch. Okay, yes. So this is the thing. Okay, you can find an equal space. This is the double helix model that you see. Okay. The plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bond, confers stability to the helical structure. So when you see that stacking, base stacking is very important. That is, one base pair stacks over the other in the double helix. Can you take the case of double helix? One base pair, okay, adenine, what does it do? It stacks over the thymine, okay, in the double helix. So this gives what? This gives stability. This gives stability to the helical structure. Clear? Okay. So here we have, after this, okay, after studying DNA, the salient features, next we are coming into an important point. Okay, how is this DNA, RNA, everything related in molecular biology? So let's learn that. So that was proposed, that is how this linkage of DNA, RNA, and how it becomes a protein, or how the genetic information in the nucleus is decoded into a particular protein, and how that protein is expressed. Okay, so that was proposed by Francis Crick. Okay, and it was named central dogma in molecular biology. So Francis Crick proposed central dogma in molecular biology, and according to this, what happens is the information, genetic information flows from DNA to RNA to protein. Okay, so we have this DNA over here. So DNA undergoes replication, that it makes its own copy. And this DNA will be transcribed to RNA by the process transcription. So through transcription, DNA will be converted to mRNA. mRNA via translation process, it will be converted to protein. So this is the central dogma in molecular biology. Molecular biology is actually surrounding this central dogma. So how is DNA replicated? How is DNA transcribed into mRNA? How is mRNA translated into protein? So everything comes under this central dogma of molecular biology. This was proposed by Francis Crick. You need to understand this. Okay, in this chapter, the rest of the content is about this transcription and translation, the process, the enzymes involved, everything. So you need to learn this. So DNA becomes mRNA through a process called transcription. mRNA becomes protein through a process called translation. Clear? Yes. So with this, we have actually covered almost the basic part of the structure of the DNA, the topic structure of the DNA. We have like almost 8 questions following this. So let's see whether we can answer these questions. Okay. So the first question is, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This statement was proposed by, for which mechanism and by whom? So what are they saying? They say that they have noticed a specific pairing in DNA. Okay, what is that pairing? Nitrogenous base pairing, that is adenine through 2 hydrogen bond pairs with thymine, guanine with 3 hydrogen bond pairs with cytosine. So this is the specific base pairing that they have seen. So they're saying that they have seen this base pairing, and this base pairing mechanism is the principle or the mechanism under copying of genetic material. Okay, copying of genetic material. So they are asking us, this statement is for which mechanism? So for copying means what we mean by copying of DNA? Copying of DNA means nothing but replication of DNA. So copying of DNA means it is replication. Okay, 1 minute. It is replication of DNA. Okay, so we can actually cancel out these two options. Next, we need to understand who proposed this. So it was not by Meselson and Stein, but it was by Watson and Crick. So Watson and Crick actually proposed this mechanism of replication of DNA. Okay, so the correct answer is option D. I hope it's clear for you. Next. So we have an assertion and reasoning statement. So assertion is, two chains of double helical DNA have antiparallel polarity. It means if one chain has the polarity 5' to 3', the other has 3' to 5'. So what do you think, students? Is the assertion a correct or an incorrect statement? It's a correct statement, right? We have already learned that the two chains in a double helical DNA is antiparallel in nature. If one is 5' to 3', then the other one is 3' to 5'. We already know that. Okay. And that is what it's explained over here. So what is it? Which option will it come? A and R are true, right? And R is the correct explanation of A. That is option D. Okay, it was an easy question. Next. So here we have almost 5 statements coming in, and you need to choose the correct statement from the options. Okay, you need to choose out the correct statement. So let's read each of the statement and find whether they are true or false. The first statement is, phosphate group linked to the OH of the 5th carbon of a nucleoside through phosphodiester linkage results in a nucleotide. Is it true or false? So here the statement is false. Why? This is the incorrect term here. It's not through phosphodiester linkage, it is through phosphoester linkage. Okay, so A statement is false. So you can come to the option. They are asking us the correct statement. So if A is wrong, you can easily ignore option A and option D because they have A in them. The next statement is, a nucleotide has pentose sugar, a nitrogenous base, and a phosphate group. That is true statement. We have already learned the components of a nucleotide. Okay. Then next one is, DNA present in nucleus was first identified by Mouse, Wilkins, and Rosalind Franklin. We told that no, They are actually discovered by Meisher. Okay, a scientist called Meisher, and he named it as what? Nuclei, right? Nuclei. So this is a false statement. Okay, so C is also false statement. So you can actually cancel out the C option also. So we already came into the option. The correct answer is B and D. So without reading these two statements, that is D and E, we can easily come and answer this question as option B, B and D only. Okay. However, we'll just simply read the left out statement. So one is the backbone of polynucleotide chain is formed by sugar phosphate. Okay, that is a true statement. Then the 5-methyl uracil is present at second position in ribose sugar. Okay, in case of ribose sugar, 5-methyl uracil is actually what? Thymine. And thymine is present at the second position, they are saying. This is a totally wrong statement. Okay, 5-methyl uracil is thymine, and thymine cannot be attached to the ribose sugar, right? It's only found in the DNA. Again, it is not ribose sugar, it is deoxyribose sugar. It should be deoxyribose sugar. Okay, so the answer is B and D, option B. Next question, again we have a couple of statements and you need to pick out the correct statement. The first statement is two polynucleotide chains of DNA are coiled in right-handed fashion. This is a true statement. Okay. The pitch of the DNA helix is 0.34 nanometer. No, it is 3.4 nanometer. Okay, so this is a false statement. The bases in two strands of the DNA are paired through glycosidic bond. It's false. It is through what? It is through hydrogen bond. Purine pyrimidine pairing generates approximately uniform distance between two strands of DNA helix. That is also a true statement. There are roughly 10 base pairs in each turn. That is also true. We already learned that, right? So the correct statements are A, B, and E. So that is option A. It's easy, right? Yes. Next one. The base pairing in DNA is specific and identify the correct group of base pairs of DNA. So base pairs of DNA means we have nitrogenous base adenine, thymine, guanine and cytosine coming in. So adenine is via two double bonds, it is paired to thymine, and cytosine via three hydrogen bond, it is bonded to guanine. So this is the correct statement. Okay, rest of it is wrong. Okay, either they have mistaken the number of hydrogen bonds. Okay. Yes. In DNA nucleotide, the attachment of phosphate group to the pentose sugar generally occurs at which all position carbon? Is it 2 prime 5 prime? No. 2 prime 3 prime? No. No. 5 prime 5 prime? It is. Yes. It's 3 prime and 5 prime carbon. That is why it has got the polarity. If one strand is 3 prime to 5 prime, the other strand is what? 5 prime to 3 prime. Is it clear? Okay, is it clear? Yes. Okay, next we have a match the following question coming in. So we have nitrogenous base, nucleotide, uracil, thymine. Here we have in the list to RNA, purines and pyrimidines, DNA, nitrogenous base, pentose sugar and phosphate group. So let's learn this. So nitrogenous bases are purines and pyrimidine. Yes, you're correct. So A is 2. Nucleotide Nucleotide is 4. So these are the components of a nucleotide. So B it is 4. Then uracil you can see in RNA. So C it is 1. Then thymine you can see in DNA. So D it is 3. So A2, B4 and C1 and D3. So that is the fourth option, D option. So this is the correct answer. Okay. So we have the next question coming in. From the given flowchart, identify the labeled A, B, and C, the concept proposed by whom. So you need to just label. So what is this, students? What is this? I hope you can answer this now. So this is the Francis and Crick's central Dogma of Molecular Biology. Okay, MD, Central Dogma of Molecular Biology, and it was proposed by Watson and Crick. So what is B? B is replication, right? B is replication. So B as replication, we have two options. You can cancel out B and C. So the answer will be either A or D. So let's see. DNA to RNA, it will be transcription. That is C, it is what? C, it is transcription. So yeah, so here we have C here as transcription. Here C is translation. So we understood that this is the wrong statement. C as transcription. Then A is what? From mRNA to protein, it is translation. Clear? So A is translation, B replication, C transcription, and this is the central dogma proposed by Francis Crick. So this is the correct answer. Okay. Yes, now we have a small topic to cover. With that, we'll be coming to the end of our session. That is packaging of DNA helix. So first we'll be learning what is the significance of packaging this DNA into the nucleus. So let's learn the basic things. Okay, the first thing is the distance between two base pairs is 0.34 nanometer. That we have already learned, right? Between two base pairs, that is 0.34 nanometer. And the total number of base pairs in case of humans. So the value given here is in case of humans, it is 6.6 into 10 raised to 9 base pairs we have. Okay, so if you multiply this, okay, if you multiply 6.6 into 10 raised to 9 into 0.34 nanometer, and you do all the conversion of this nanometer into meters, you get how much? 2.2 meters. Okay, so 2.2 meters means it's very long, right? So the whole DNA is like almost 2.2 meters, which is very long. So we need to accommodate this long DNA into the nucleus. So there comes the significance of packaging, right? So in DNA, in prokaryotes, that is examples include bacteria, they don't have a well-defined nucleus, right? So that doesn't mean that DNA is scattered all throughout the cell. Okay, DNA, we told, no, they are acidic in nature and they are negatively charged. These DNA are held with some proteins. Okay, they are held with some proteins and they are held together in a region known as nucleoid. So if you have a prokaryotic cell here, they don't have a well-defined nucleus. So they have a nucleoid region where you can see the DNA, which is held with some proteins. Okay, and this DNA nucleotide is organized in large loop which is held by proteins. Okay, so this DNA in the nucleotide is organized into large loops. Okay, and they are held by proteins. Okay, now if you take the case of eukaryote, okay, how this packaging is done in eukaryote, it is with the help of positively charged basic proteins. That means we told that our DNA is negatively charged. So DNA will be linked to positively charged basic proteins and they are called histones. Okay, so DNA will be linked to positively charged proteins called histones, and histones are rich in basic amino acids like lysine and arginine. Okay, so these are the names of two amino acids. Histones are made up of these two amino acids and they are basic amino acids. Basic amino acids means their side chains. That is, the amino acids side chain consist of positive charge. Okay, they have what? They have positive charge. Okay, histones are organized to form a unit of 8 molecules called histone octamer. Okay, so that is actually the structure of the histone. So it's like a football. You have a ball and it is made up of 8 molecules. It's organized to form 8 molecules and it is called what? Histone octamer. Okay, is it clear? Right. So let's see. The negatively charged DNA is wrapped around the positively charged histone. So we studied the histone or the protein to which the DNA is attached is positively charged due to the presence of the basic amino acid lysine and arginine. Okay, lysine and arginine. And When this negatively charged DNA is wrapped around this octamer, it forms a structure called what? Nucleosome. It forms structure called nucleosome. So here you can see the structure of a nucleosome. So this green part is the histone octamer. To that the DNA is wrapped around, and this is again one component of histone. We have different types of histones. Okay, we have H1 histone, H2A, H2B histone, H3, H4 histone. So here we have the H1 histone. So we have this DNA which is wrapped around the histone octamer, and they form a structure known as nucleosome. So a typical nucleosome consists of 200 base pairs. Okay, a typical nucleosome consists of what? It consists of a 200 base pair of DNA helix. So you have a DNA helix which is bound to the histone molecule, and how much it has, how many base pairs it has? It has 200 base pairs. It has 200 base pairs of DNA helix. Okay, and the nucleosome constitute thread-like structure in nucleus. So we have this DNA which is bound to what? Which is bound to a histone octamer. So when you look through an electron microscope, it will be like a bead on string structure. So here we have an electron microscope imaging. So the whole packaging will be like to one bead what is arranged. You have a DNA molecule wrap and it is connected to next histone. So it will be like a bead on string structure when you observe it under the electron microscope. Okay, so this bead on string structure we call it as chromatin. Okay, so chromatin will be a thread-like structure or a bead on string structure, and we call them as chromatin. And this chromatin is packaged to form what? Chromatin fibers. Okay, chromatin is packaged from chromatin fibers, and these chromatin fibers are further coiled. Okay, and they are condensed at metaphase stage of cell division. So when these chromatin fibers are further coiled, we call them as chromosomes. Okay, so don't get confused with what chromatin is, what chromatin fiber is, and what chromosome is. So basically, we'll be having the chromatin first. Okay, the DNA will be packaged with histone to form chromatin. Chromatin will become what? Chromatin will become chromatin fiber, and the chromatin fiber will become what? It will become the chromosome, that is highly condensed chromatin fiber. Okay, and this chromosome will be, we can find this chromosome in which stage of cell division? In metaphase stage of cell division. So that is when it's fully condensed, the cell is going to divide and they start to align at the metaphase plate. Okay, so the packaging at higher level, that is if you are intending to package it higher level, like higher level of packaging isn't when it comes to the level of chromosome level. Okay, so this higher level of packaging will require additional proteins, okay, other than histone molecules, and they are generally or commonly referred as non-histone chromosomal proteins. Okay, or we call them as NHC. Okay, NHC proteins. Okay, MHC proteins, if they need to be additionally wound. Okay. Okay, so based on this condensation or based on this packaging, we can actually we can classify the chromatin as heterochromatin and euchromatin. So euchromatin means it will be loosely packed. Okay, the chromatin will be loosely packed, and it is referred to as euchromatin. And this euchromatin is transcriptionally it is active. Okay, euchromatin is transcriptionally it is active because it is loosely packed, right? It's loosely packed and it will be transcriptionally active. Now the chromatin that is more densely packed, the chromatin is more densely packed, and if it is stained dark, it is heterochromatin. Okay, so one type is euchromatin and the second type is heterochromatin. That means it will be densely packed, it will be stained dark. Here, in case of euchromatin, it will be loosely packed and it will be lightly stained. And it will be lightly stained. Okay, and this will be transcriptionally inactive. So this will be transcriptionally inactive. Which one? heterochromatin, because if you ask me what is the reason why they are transcriptionally inactive, because heterochromatin means they are condensed, right? They are condensed. So transcription factors or enzymes cannot enter, okay, and start the transcription. But in case of euchromatin, the DNA is completely in a decondensed form, it is loosely packed. So transcription factors and polymerase can easily enter and they can bind to the initiation sites and start transcription. So that you will learn a much more in a better way, or in a more clear way when we discuss transcription, right? Okay, so that is all about packaging of DNA. Okay, so basically you have to study what a histone is, how DNA is wrapped around histone, how many base pairs of DNA is wrapped around histone. That is almost 200 base pairs of DNA. Then the distance, and then the histone is made up of basic amino acid like lysine and arginine. Then the euchromatin, heterochromatin, everything. Okay, so I hope the session was clear to you. So in this session, we basically dealt with the structure of the DNA, the polynucleotide chain, then the structure, the polarity, the different components of deoxyribonucleotides, everything. So I hope everything was clear to you. Okay, so if that's it, then we'll end the session. So thank you, everyone. See you soon with the next session. Okay, bye.